Let's go live. Everybody say hello.
Hello.
Thank you.
Almost. We're actually on this time.
What's on the back? What was that one?
Let's talk about the Nobel Prize, the 2020.
Oh, for CRISPR-Cas9?
Like the genetic scissors.
Yeah.
Yeah.
It's called CRISPR-Cas9.
It's pretty cool.
Donna is the, what's your first name again? Allison? Yes. What do you want to know about it?
Oh, we just looked it up.
Yeah. It is a way of directing a genetic mutation or a genetic change, but you. All right. So we've been talking about viruses, and we finished up with kind of a couple of the different kinds of viral life cycles, in a sense, in that viruses can operate in a couple of different ways. They can get into a cell and begin assembling themselves and replicating, and then pop the cell open. Or they can get into the cell and actually take their genome and incorporate it into the chromosome, and as the cell divides, the replicated cells are also carrying the virus. And so this is known as the lysogenic cycle. And it's an important vocabulary term distinction. The lytic cycle or the lysogenic cycle. And these come about in part from our work with bacteriophage. You can have a bacteriophage that is simply replicating and going to blow the cell apart. You also have some phage that are incorporating into the genome of some of the bacteria. And in the phage biz, you actually end up with this plaque that we were looking at. That's the way you determine if you have a virus. The plaque can be completely clear, meaning that all the bacteria that were infected with the virus popped. Sometimes the plaque can be a little bit cloudy, meaning that there's some bacteria within this sphere of being infected that didn't pop. They actually have the virus in them as what's known as a stable lysogen. And happily, most viruses don't lysogenize within us. But there are a few, in fact, like herpes, that actually does lysogenize. And it ends up, in a sense, being carried, and you have this latent form of it. The chickenpox virus does this as well, which is how you get shingles. So we finished up here in that we've been talking a lot about antibiotics, and antibiotics are an absolute miracle because bacterial infections can absolutely be devastating, but with antibiotics, they really are just no big deal. Viruses, on the other hand, as we are living through it at the moment, are a much more difficult thing to treat. There are very few antiviral drugs. So antivirals do exist, but they are going to be specific for certain viruses. I mean, antibiotics are specific for certain bacteria, but it's very broad classes of bacteria. But antivirals are often very specific to a virus. Additionally, because a virus is not a free-living thing, you're going to have to start acting on either blocking its uptake into the cell, that's not a bad way to do it, or inhibiting its ability to replicate within the cell. And that can be a pretty difficult thing, given that it's actually using your very own cell machinery. And so a couple of things you can do is you can block the ability of a virus to get into the cell. And you may have heard of Tamiflu. This is a drug that is given when people start to show signs of the standard flu, and it can actually reduce the longevity of the disease. On a personal note, I've taken Tamiflu, and I didn't think it did jack. And the company had an initial ad campaign with the Olympic diver Greg Louganis. I'm not sure if anybody knows it anymore. But he was talking about how, yep, in my training I had to keep my health up, and I had the flu, began to feel the symptoms. I took Tamiflu, and I was back at it, in 2 days. And it's like, dude, that's not how it works. Even in the directions in the medicine itself says that it will reduce the length of symptoms. So if you have the flu and it would normally be about 10 days, taking Tamiflu will reduce it to about 6 or 7 days. And I don't know when the last time you had the flu, and I haven't had the flu in a long time, but I usually feel pretty lousy for a day or for 2 days, and then I feel like hell for a couple of weeks, but what the hell, or a week anyway. And it's not like I'm debilitated in bed. And I've not said this to the world. I don't want to get sick. I didn't get the flu shot, whatever. I don't want to talk about that. I don't find it very effective. All right. We have some pretty good successes, though, with HIV. There is a drug cocktail that can block the replication of the virus that causes AIDS. And if left untreated, the virus that causes AIDS begins to destroy your immune system. It begins to affect your T cells, which are part of your immune response. And as your T cell count drops, your ability to fight off a whole bunch of other things is dramatically decreased. And so you end up having fairly weird cancers that normally your immune system would take out, and pneumonia, all kinds of bacterial infections and yeast infections, and you are subject to all kinds of things. It's a way of seeing just how critical your immune system is. So there are drugs that you can take that will inhibit the replication of the virus, allow your T cell count to get back to normal, and as long as you maintain them, you are going to be actually doing well. HIV is one of these that actually does form latent sort of hiding spots in different parts of the body, in different organs, and even in the brain. And so it's very difficult to completely remove HIV from a system, from the body. Another one that you may have heard of is acyclovir. And I'm trying to think what the trade name is. Abreva, maybe. There's like these little teeny toothpaste tubes that if you've got a cold sore, a herpes sore, you can put a little drop of it on there, and it works pretty well to lessen the outbreak. It'll shorten up the time frame. And this is an inhibitor of a DNA polymerase. And so, the DNA polymerase for the virus is not, in this specific case, is not the same as the normal DNA polymerase in the body. And so it's more sensitive to picking up strange things, or it can be inhibited in this way. With HIV, the drug AZT is able to inhibit part of the viral life cycle, and it's hitting an enzyme that isn't naturally occurring in the body, and that enzyme is not less stringent about what kind of nucleotides it picks up, and so it'll pick up some of these strange nucleotide analogs, and it works. How about this, though? Vaccinations are probably our best course of action for viruses. With all of these things, you get the virus, you begin to show symptoms, and the best you're going to be able to do is reduce the longevity or the length of time that you have the virus, or put the virus into some sort of latent mode, but you're not really getting rid of it with a lot of things, or you're simply shortening it. You still have a pretty good viral infection. Vaccinations are a pretty cool trick. So we are not defenseless, happily, against viruses, in that coffee moment. In the same way we have all these great natural defenses against bacteria, we actually have similar defenses against viruses. Our epithelium does a really good job. In part, the very top of your cell is dead cells. Dead cells can't support a virus. The viruses that get into our bloodstream, there are a whole range of different sort of defense molecules, things like proteases. They are always floating around. They can begin breaking down foreign molecules. There are inside the cell a range of nucleases that are going to actually attack the invading DNA and RNA. There are a whole host of inflammatory and antiviral type systems. Interferon, you may have heard of, is a protein that is being released by infected cells and is essentially saying, hey, we got a virus in the system, let's start doing something about it. There are parts of the immune system like leukocytes, white blood cells. These are able to destroy cells that show some signs of being infected. When the virus hits, it's going to either uncoat or inject its DNA and leave its protein coat. That protein coat is going to be on the cell surface still. And so the immune system is able to pick that up and say, you have been infected. And that cell can be targeted for death. We also have immunity that allows us to, if you've seen the virus before, you can remove it from circulation before it even gets into cells. You can begin to remove the virus or cells that have been infected from the virus very quickly. And so the immunity is pretty much your number one goal. One of the points about this is maintaining a good immune system is one of the best ways you can prevent viral infection. And how do you maintain a good immune system? Get enough sleep, eat well, less stress. Those are really critical things that help to keep us from having virus diseases and viruses getting a foothold. So interferon has been a possible general antiviral. And interferon acts to turn on a whole host of antiviral proteins, and it's a way of establishing that a cell has been breached. So interferon is being generated by cells that have a virus inside them, and then it in turn can affect other cells and get them sort of activating their viral defense systems ahead of time. So there are a couple of different ways that we can actually begin to stop a virus. And this is just some general notions here of different places where interferon is able to actually block different viruses. These are typically viruses that when they infect a cell, we can begin to activate systems that will block their further replication. So it's not really blocking the cell's ability to, or the cell's uptake of the virus. It's going to end up blocking the viral reactions going on. And we don't need to get into that too much. So viruses have a whole range of effects. Viruses can cause cell lysis. So viruses that are in the lysogenic cycle, or in the lytic cycle, can cause cells to absolutely pop and rupture. Viruses can serve as a factory for producing more viruses, and that weakens the cell's ability to carry out its normal metabolism. You end up with a variety of different kinds of effects here. One of the things that viruses can do is transform cells into cancer cells. And this is actually a sort of the introduction to cancer biology in general, which is a really cool area. And it's just not what the class is about, but it is one of my areas of intense interest. and that is oncology and cancer biology. And we are learning more and more about the connection between cancer and viruses. And let me just take a moment here. Yeah, let's do a little bit of board work and then we can look at this. Because this is an area that is pretty darn neat. So, how many of you have kitty cats? And your cat has probably been vaccinated against feline leukemia virus.
All three of them.
Yes. Feline leukemia virus is a virus that is transmissible, and rather than giving you the sniffles, it causes leukemia, which is the uncontrolled replication of cells in the bloodstream, of the white blood cells. And so, at the moment, we don't have a viral system in humans that is as direct as things like feline leukemia virus. That is, a virus that, were you to catch it, actually would begin causing a cancer. The human papillomavirus is close, but even that is not a direct shot. It's sort of an ancillary point towards getting different kinds of uterine cancers and things. So that's a really, a lucky thing for our world, is that we don't have these kinds of cancer-causing viruses. They're present actually in a lot of vertebrates. Mice have them, chicken have them, cats have them. Dogs don't have them. Monkeys have some of these. And there are viruses that cause muscle tumors, there are viruses that cause leukemia a lot. There are viruses that cause a whole range of different kinds of cancers. So there are a number of parts to this that are really pretty cool. And while we don't have direct transforming viruses in the human population, it's not out of the question that we could generate them. And we do have an increasing number of cancers that are virally associated. First off, if we start thinking about cancer mechanisms, we have this notion of what's called the transformed state. And these are cells that are not behaving properly. They are acting like cancer cells. And there's a couple of hallmarks to the transformed state. Part of this has to do with the way, if you're dealing with cell culture, the way cells behave. I mean, that's a lot of where the hardcore biochemistry of this starts to come about. And so it's a way of assessing whether a cell is cancerous or not. So or maybe not, if you have a symptom, a lump, you have a fine needle biopsy is often something that's done, in which you actually target the tumor, pull some cells out. And one of the things that is done is a cytologist or a trained pathologist will look at the cells and begin looking for the amount of membrane they have and irregular shapes to the cell. And there are certain cues that a good trained pathologist is able to look at and say, okay, this is abnormal tissue. The same thing happens when you're getting a pap smear. And it's one of these things that I find as a molecular biologist, I'm like, man, the pap smear is not a whole lot more complicated or different than our gram stain technology. And it is something that's been around since the middle of the 19th century. But it's a really good screening tool for looking at cell morphology and cell shape and the membrane accumulation. So it's still done. But we also have a whole range of molecular diagnostic tools as well. So the transformed state, from a cancer biology perspective, there are a couple of things that indicate cells have gone off the rails. And so what you might do with a fine needle biopsy, in addition to having the pathologist look at it, is you put it into culture. You actually start to grow it up in a dish. A couple of things that indicate something's funny. One is what's referred to as a loss of contact inhibition. And this is something that eukaryotic animal cells exhibit, in that you have a tissue culture plate and you're growing the cells up on it. And you can do this by what's called primary cell culture. You scrape the inside of your cheek, put it into cell media. You got to add a lot of antibiotics because there's going to be a lot of bacteria. You knock those back. The cells begin to divide. They divide about once every 24 hours. And after a week or so, you'll have the cells covering the plate. Now, if you take a little spatula with a little rubber tip and go and scrape along a stripe, the cells will begin to grow back into that stripe. And the same thing's happening if you cut yourself. That's how the cells actually are growing back into that area. Now, normal cells that are not transformed, when they touch, they actually make cell-cell connections that say, yo, you've reached the other side, stop dividing. But transformed cells will hit another cell, and rather than stopping dividing, so we have this gap here, we got division and division, and everybody's dividing, and beep, we hit. A transformed cell keeps on dividing, and then it'll actually start to pile up. And that is an indicator of a tumor. Another part of the transformed state, and this is an old-fashioned term, but you actually still hear it, it is referred to as the ability to form foci in soft agar. Foci are the term that in micro we call it a colony. This is going to be a collection of cells. And soft agar is, as the name implies, a little bit like our gelatin stuff. It is not nearly as bouncy as our agar plates. Soft agar is a goo. And standard cells, animal cells, human cells, they don't grow unless they've got a surface to sit on. Bacteria cells, as we know, will grow in suspension really, really well. Bacteria cells, they absolutely have the ability to form foci in soft agar. Human cells ought not. Animal cells should not. So if they are growing in the sense they can grow in liquid culture, they don't have to have a solid medium, that's another sign of the transformed state. And so this is how we begin to assess the transformed state. One of the critical experiments that begins linking viruses to cancer was back in the late 70s, early 80s. Bishop and Varmus began doing some experiments. They were looking at a variety of different viral systems that cause cancer, and they were looking at, I think, a chicken virus, but I can't remember exactly which one it was. Regardless, they've got a transforming virus. And they can infect cells with this transforming virus. The cells go from being normal, contact inhibited, requiring a plate to grow on, into becoming transformed cells. And they do this by infecting them with the virus. What they did was begin breaking the virus up. I think it was like the chicken erythroblastosis virus, but that's not important. They began breaking the virus up into little pieces of DNA. And what they identified were specific chunks of DNA that were actually carrying out the transformation. They isolated these pieces of DNA, and they found, well, what they called this was an oncogene, a gene that would cause cancer. Now, the really amazing thing was when they began comparing the oncogene from the virus to genes in the chicken, they found that there is, in fact, a non-cancer-causing normally in the genome form of this gene. And so we've got what they refer to as the viral oncogene. And while it's not exact, it's very clearly a part of and very similar to a cellular form, the cellular oncogene. So in the cell, this gene is being controlled very carefully. And often the virus has a part of the gene, not the entire thing. So it may be missing a regulatory portion of the protein. The virus also might have a portion of the gene, or it has the gene, but it's not under a real strict control that's on all the time. And these oncogenes are typically the genes that will trigger cell division. They're going to be involved in saying, hey, it's time to start dividing. And they are generally, the cellular form of it, is under real tight control. You don't express them at high level. They have a series of phosphorylation events they have to go through. I mean, it's like we don't want to have these things on just sort of randomly because they cause a cellular division. Don't do that. The viral form of it doesn't have the regulation. It's on all the time. It's a little bit like somebody is just ringing the doorbell all the time. And over time, it makes people crazy. It is going to cause the cell to go into this abnormal division. So Bishop and Barmas developed what they called the V-onc and the C-onc. The viral oncogene and the cellular oncogene. And this led to a whole discovery of a class of genes within the animal genome that are known as oncogenes. They are genes that, if disrupted, can cause cancer. And there are cancer-causing viruses that have, over evolutionary time, landed in the genome. So let's just say here is a piece of DNA, and here is some kind of signaling protein. And it's got a really potent regulatory region here that keeps it from just turning its light on all the time. A virus can land near the genome, or near the spot. So the virus lands in here, and the virus can land, and when it begins to pull itself back out, rather than just pulling just this part out, it pulls out a whole mess of stuff. And so you now have the authentic virus plus a bit of the signaling regulatory piece. And this just happened randomly. The virus landed here. It had probably picked up over its life cycle a bunch of pieces of DNA, but this one actually has an effect when it gets into another cell. And what this can do is essentially take and put in this mutant copy of a regulatory gene. Mutant such that it's on all the time, it's causing the cells to divide all the time, and that's how you can actually have a transmissible cancer-causing virus. So a class of viruses that do this a lot, that is, they integrate and then they pull out more than what they should, are viruses known as retroviruses. These are RNA viruses, and in order to actually carry out their replication, they do a really weird thing that is actually sort of backwards to this flow of information that we know of. They are going from an RNA molecule, and they generate a double-stranded DNA molecule. Now, if you think back on this, we have gone through in our genetic stuff the idea of what we call the molecular dogma, the flow of information, and that is DNA is transcribed to RNA, RNA is translated to protein. DNA can replicate itself, but RNA never goes to DNA. That is just flat-ass weird. You don't do that. Viruses have the ability to do that. Some do. Retroviruses do. And it requires a very specific enzyme known as reverse transcriptase. Now, the official sort of enzymatic term for reverse transcriptase is an RNA-dependent DNA polymerase. And if we kind of break that apart, a DNA-dependent DNA polymerase is replicating DNA all the time. That's easy. Everybody's got those. A DNA-dependent RNA polymerase, that's what makes RNA. We all got those. An RNA-dependent DNA polymerase, that's just flat-ass weird. Ain't nobody got those. Viruses do. And so what the retrovirus life cycle is, it comes in as RNA. It can then convert itself to DNA, and that DNA is what gets into the cell, or into the genome. And this is kind of a cute trick if you think about this. What the virus has done is it comes in as RNA. The RNA is something that is translatable. It can begin to produce proteins. One of the proteins it produces is reverse transcriptase. That in turn turns the genome into DNA, and then this DNA makes its way, as all DNAs do, into the nucleus, and it integrates into the nucleus. At that point, it is you. It is part of you. You own it. And you can't get rid of it. And now, if this virus wants to replicate, it's acting like a gene in the genome. All you do is start producing RNA, like all good genes do. But in this case, this is in fact the viral genome. And your cells are now producing the virus using your own machinery. I mean, it has absolutely hijacked your entire world. And the virus now is able to replicate. The virus begins producing proteins. The virus lyses the cell, and you're off to the races. Additionally, you might have a retrovirus that integrates into the genome, and in addition to the viral proteins, it also is producing some kind of mutant cell signaling protein that is effectively causing your doorbell to ring. I kind of like that analogy, actually. If you think about an old-fashioned doorbell, not a Bluetooth-enabled one, you got the button on the outside of the door. Dude comes up and pushes that button. That activates a switch inside the house. That wire goes down in the basement, hooks to a bell, and that's how your doorbell works. You got a couple of different parts. You got the guy on the outside pushing the button, and then you had the signal on the inside ringing. What if you were to have some kind of short circuit in which ain't nobody pushing the button, but it's ringing like crazy all the time inside? That's what these mutant proteins are doing. They don't require an external button pusher. They're just on. And this is what causes cell division to just go haywire. It just starts cell dividing really, really fast, and you begin to generate a malignant tumor this way. So viruses can do a range of things. Let's just look for a moment at a couple more figures. But viral biology and oncology are kind of amazing systems that are a fallout of the complexity of the nature of life on this here planet. All right, so this is a way to think about this, is that if you want to replicate as a virus, you have to have the cells going into DNA replication. But our cells, for the most part, aren't always in DNA replication. So what does the virus do? It triggers DNA replication. And that's why, the virus doesn't have an intention. It is just the evolutionary mechanism of how it works. We can't say that the virus is evil. The virus is just doing what it does, which is to make more of itself. It can't help itself. The coronavirus has evolved and developed in a way because it is really good at replicating itself. That's its own motivation. Making people sick is not something the virus is actually intending to do. It just wants to make more of itself. So the viral proteins that are introduced are going to trigger the DNA cell cycle. Now, one of the difficulties is if you've got a viral infection that is putting in one of these always-on cell signaling proteins, that's how we have a transforming virus. And the human papillomavirus is one that can do this. So human papillomavirus, it is not a direct, but it is a pretty good trigger for the development of ovarian tumors and uterine or cervical cancer, cervical tumors. So in this case, we have infection by HPV. It infects epithelial cells in the cervix. The DNA of the virus integrates into the cellular genome, and those infected cells begin to carry out viral replication, and then over time, they can probably make the cell a little bit more sensitive to other kinds of mutagens and make it so that exposure to all the stuff we have in our environment is you're now more susceptible to it, and you can begin to get cervical cancer. So it's a fairly low percentage, but it is one of the first viruses that we can really put a finger on and say, this virus is associated with this specific cancer. So how do we deal with this? There are a bunch of HPV-type viruses, and they are associated with cervical cancer, 70% of them. And so we treat this by a vaccination. All right, we're going to just pick back up on this on Monday, but this is another kind of cancer-causing virus, these retroviruses that we talked about, and that here's a list of the viruses, and these are the kinds of names and things, the different species, the kinds of tumors, and these are the oncogenes that they have picked up and mutated. And it's one of the sort of interesting things about the way the science works is that from a backwards perspective, all these oncogenes were discovered through looking at viral oncogenes. And so you find viruses, and then the gene that they're carrying, there's going to be a homolog in the human or in the animal genome, and pretty much, in all animals, they've got these different kinds of genes. All right, we'll pick back up on here and be scared of eating hamburgers on Monday, at least rare. No, actually, don't eat cow brains over the weekend until you can talk about things. Avoid cow brains. In fact, in general, avoid eating brains. If you're faced with a cannibal wants to offer you some sort of ritual meal, go for muscle, go for heart, don't take the brain. I leave you with that wisdom.